All right. So when we talk about our homeostasis, right? So what is homeostasis? Homeostasis is basically the maintenance of an internal body environment. Maintenance of an internal body environment is referred to as homeostasis, right? So this will include everything. When we talk about homeostasis, we mean everything. I mean, oxygen concentration in your body. It's maintained within a certain range, right? If it becomes lesser than that or more than that, the body will try to bring it back to normal. Your carbon dioxide concentration in the blood, pH of the blood, uh, the amount of glucose inside the body, the temperature of the body, the minerals inside the body, sodium, potassium, calcium, all these mineral ions, right? They are all, what are they? They are all maintained by the body within a certain range. So that maintenance of the body conditions is referred to as homeostasis. Now, one of the key organs that helps in maintenance of homeostasis is obviously your kidneys. Okay. Now, when we talk about excretion, okay, so there are a few different excretory organs in your body. I know we don't realize it, but there are quite a few excretory organs in our body other than the kidneys. For example, we have our lungs. From lungs, water vapor is also released. In the lungs, water vapor is also excreted. And most importantly, carbon dioxide is excreted or removed from the body. Then another really important gland that we have is our sweat. Sweat gland also is involved in excretion. It removes water. It also removes mineral ions and a few other substances as well. And then, of course, we have our kidneys. Kidneys are important for the excretion of water, urea. Urea is basically your nitrogenous waste products, right? Urea is considered as a nitrogenous waste product. Urea forms in a process that is known as deamination. What is deamination? Deamination is breakdown of amino acids by the liver, of excessive amino acids by the liver. So in the process of deamination, basically urea is the one that is produced as the waste product. Then salt, sodium chloride, potassium, phosphate, ammonia. These are all excreted from the body with the help of kidneys. When we talk about our so when you talk about the kidney, The kidney is involved in both. Kidney is also important for hemostasis and it's also important for excretion. How is it important for hemostasis? It helps maintain the mineral ion concentration within the body and keeps it within a specific range. Now, so let's go ahead and take a look at the topic of kidneys and let's study about exactly what the structure of kidney is like and how do the kidneys function. First, let's take a look at the general urinary system overall, and then we will take a look specifically at the structure of a kidney. Give me a minute. I'm going to share a diagram with you. That's going to be very helpful. Hold on. All right, so our urinary system is composed of our kidneys. So there are two kidneys. You have left kidney and you have right kidney. Both the kidneys are supplied with the help of renal artery. Renal artery is a branch of aorta. Right? And the blood that leaves the kidney through the renal vein, which eventually joins up with vena cava. Then we have two kidneys, which is left kidney and right kidney. The left kidney and the right kidney is going to drain through the ureter. So ureter is a tube that connects the kidney to the bladder. And the bladder is a muscular structure that is responsible for holding and storing urine. So the kidney, what is going to happen? The kidney is connected to the bladder through the ureter. In towards the exit of the bladder, we have sphincter muscles. The sphincter muscles help to control the opening of the bladder, and the urine will exit the bladder through the urethra. Right? This is the general structure of how your urinary system is. Now, in your urinary system, you can specifically see that we're talking about kidney. So we are going to study, number one, we'll study about the structure of kidney. Then we will study about the structure of the cell of a kidney. Okay. So we're just think of it this way, that you're going deeper, deeper, deeper into the tissues. Let's take a look at the structure of kidney. 1 second, let me share the diagram with you. Okay, so in the structure of kidney, you can see that we have the outermost layer, which is your fibrous capsule, and then we have two layers inside the kidney. So we have the dark staining area and we have the light staining area. Okay, the light staining area is referred to as is the sorry the dark staining area is on the outside. Okay, the darker staining area is on the outside. We call it the cortex. Okay, then we have our lighter area. The lighter area is referred to as the medulla. Okay, and the medulla has these pyramids inside it. See, there are pyramid-like structures. So this is your, we just call it the pyramid of medulla. That's it. So the darker outer layer is the cortex, and the lighter inner layer is going to be referred to as the medulla. And what actually happens is that why do we see something like this? The reason we see something like this is because the cortex is rich in capillaries. Where I go? Thank you. Hold on. I'll try again. It is rich in capillaries, so it tends to have a darker red color. When we talk about our medulla, so the medulla drains into a structure that we call the renal pelvis. Okay, the medulla drains into a structure that we call the renal pelvis. So the urine basically forms inside the cortex and the medulla, and once the urine is formed inside the cortex and the medulla, it travels to the pelvis, and from the pelvis it goes to the ureter, and ureter will then carry it to the bladder. Now we are going to go zoom in even more. We literally imagine like we're zooming deeper and deeper into the tissues. So now let's zoom inside the kidney and let's go down to just one single cell, right? What if we take just one single cell of the kidney and we look at that? So that is the one single cell of a kidney is referred to as a nephron. So one single cell of a kidney is referred to as a nephron, right? Like a cell of the liver is referred to as hepatocyte. Your cell of the wall of the blood vessel is referred to as endothelium. So just like that, the cell of a one single kidney or one single cell of a kidney is referred to as nephron. Now let's take a look at what does the structure of a nephron look like. 1 minute, let me share the diagram. All right, so this is the structure of your um, nephron. Okay, so a nephron is, if you look into it, the nephron starts with a small capsule, and inside that capsule you have like a collection of capillaries, basically. Okay, then so this capsule is basically referred to as Bowman's capsule. Okay, this is referred to as the Bowman's capsule. So what we can basically see is this, that the Bowman's capsule has the collection of blood capillaries inside of it. Then the Bowman's capsule continues on, forming a tubule. You don't have to learn the name of the tubule. It's known as the proximal convoluted tubule. Proximal means close. Convoluted means that it's, like swirly, like it's not straight. It's bending a lot. And but you don't have to learn that. It basically forms a tubule, and that tubule gives us travels further on, and it gives us a loop. Okay, this loop is referred to as loop of Henle. And the loop of Henle then continues on to the, again, we don't have to learn the name, but we call it the distal convoluted tubule. Distal means further away. Convoluted means obviously that it's all twisty instead of being straight. The tubule then continues on into the collecting duct. The collecting duct is then going to continue on to the ureter. So this is how the structure actually works. Now, when we talk about, this is what the structure of a nephron is. This is not how it works, but this is what the structure of a nephron is. Now, when we talk about how exactly does a kidney work, okay? So the functioning of a kidney can be divided into two major stages. The stage number one is ultrafiltration. And stage number two is selective resorption. What is ultrafiltration? In ultrafiltration, basically what happens is that we have our Bowman capsule. Okay. Inside the Bowman capsule, we have a collection of capillaries. The collection of capillaries is known as this collection of blood capillaries is known as a glomerulus. Okay. So what is going to happen, that the capillaries, right? Inside the capillaries, the blood is at a comparatively, it is at a higher pressure. So because the blood pressure in the capillary is high, what happens? That the fluid from the capillaries is forced out. Basically, everything other than the cells, the everything other than the red blood cells, other than your platelets, other than, large proteins as well. Okay, large proteins, red blood cells, platelets, they're all going to be left behind. Everything else, the fluids, the fluid containing hormones, the fluid containing toxins, the fluid containing glucose, the fluid containing sodium, chlorine, potassium, calcium. It's just going to squeeze out. Okay. So fluid from the capillaries is forced out, and the fluid is filtered by the basement membrane. So the red blood cells, platelets, proteins, they will not be allowed to pass through. Right? And then what is going to happen? So what is the basement membrane? Let me go ahead and let me show you. 1 second. Let me draw this out. Hold on. So basically, basement membrane is the layer between the cells of the Bowman capsule and the cells of the capillary wall. Okay. So if you look at the cells of the capillary and the cells of the capsule, so between the two of them, we have the basement membrane. The basement membrane helps in filtration. So the large molecules like large proteins, platelets, RBCs will stay back, while the fluid portion containing the hormones, containing glucose, containing water, containing your mineral ions is going to squeeze out of the capillaries. This filtrate is referred to as glomerular filtrate. And this process is referred to as ultrafiltration. So ultrafiltration, basically what are you doing? You are removing large molecules, and everything else other than those large molecules is all going to enter where? It's all going to enter into the nephron, into the capsule for further processing. So this is the first step of how a kidney works. The second important stage that we have is referred to as selective resorption. Okay, so when we talk about how exactly does a kidney perform selective resorption, let me give you a basic idea. Okay, before we go with the actual theoretical portion of it, let's go with the basic concept. The basic concept is this, that we have, let's say, we have done ultrafiltration in which we have taken all the small molecules, the fluid and everything, and we put it inside the kidney. Now, the issue is this, that everything is inside the kidney, including a lot of our mineral ions, including a lot of the salt, including a lot of the water. The body doesn't want to get rid of everything, right? The body doesn't want to get rid of everything. The body only wants to get rid of what is waste and what is excess. That's it. The body doesn't want to get rid of everything. So now the body has a very specific role. What is the specific role? The body needs to now choose which things it needs to take back. So now the body is going to run a checkup. Okay, listen, we do not have enough water inside the body. Take it back. The water will be absorbed back into the body. We definitely need to take, the glucose is always absorbed. Unless a person is diabetic, they have too much glucose in the blood, it's always taken back. Glucose is always going to be absorbed back. Minerals, sodium, potassium, chlorine, calcium, they are most of the time, they are absorbed back. If they're excessive, if there are too many in the body, then the body will not absorb them back. But if there are not enough in the body will absorb them back. So so what is going to happen? Slowly and slowly and slowly, your urine is going to become more and more concentrated, right? Initially, it's dilute with a lot of water, a lot of mineral ions, glucose, all of that, and slowly, the body is going to start absorbing all these things back. So the urea, the amino, the nitrogenous waste, the ammonia, that will all start to become more and more and more concentrated. So this is selective resorption. It's sort of in this way that imagine that you have a drawer. Okay, that drawer is very dirty. You want to clean it out, like you want to reorganize it. So what do you do is that you first dump out all the contents. Okay, except for the really, really big ones. Maybe, like the really big ones, you'll dump out most of the contents. That is ultrafiltration. Then from that, you choose that, oh, this thing I want to keep safe. I will keep this thing. This thing I want to get rid of, throw it away. That is selective absorption. So ultrafiltration is when the kidney will filter out all the fluid and everything from the blood, and then in selective resorption, it will now choose these things will take back into the body. This thing will let it stay and will let it be removed from the body. So that is what selective resorption actually is. Now, when we talk about, so basically this is all there is to selective absorption. I will go over it in a bit more detail, but that is all there is to the top concept of selective absorption, right? For this, let me go ahead and share us some image with you. Hold on, give me a second. Okay, so see, this diagram is showing you that your glucose, amino acids, proteins, vitamins, they are absorbed in the first tubule. Okay. Then after the first tubule, we get the loop of Henle. So in the first part of the loop of Henle, the part that is going down, okay, that is where water is absorbed. And the part of loop of Henle that is going up, that is where sodium, potassium, and chloride ions are basically absorbed. Then we have our second tubule, which is the distal tubule. You don't have to learn the names, just learn the second tubule. So now this is where sodium, chloride, hydrogen chloride, water, ammonia, potassium, hydrogen are all going to be resolved. Now, by the time you reach your collecting duct, okay, so now in the collecting duct, what is going to happen that you will now have a very concentrated urine, right, where all of the excessive stuff has been removed and all the excessive stuff have been removed, and you now have a very concentrated urine that is composed only of excessive glucose or excessive mineral ions or excessive water. And of course, then you have most of your used up hormones, your nitrogenous waste products, urea, ammonia, all of that, right? So when we talk about our, so this is what selective resorption is. Okay. So in selective resorption, excuse me, glucose, water, mineral ions are absorbed back in the body. The urine concentrates, the urine concentrates, and the concentration of urea plus waste products plus any excessive substances is going to increase, and then all of it will concentrate all the way to the collecting duct, and the collecting duct will connect it to the ureter, and then the body is going to go ahead and it is going to remove it. So let's say that we are doing an experiment. Okay, we are taking samples from different parts of the nephron. So what will we see? The first sample that we're going to take is going to be from the blood. Okay. First sample that we're going to take, let's take a sample from the blood. So this sample has let's say that the concentration of protein is A. Glucose, because this is blood, right? So everything will be as normal. Urea is C and sodium ion is D. Right? Now, what is going to happen? Let's take another sample. This time, let's take it from the Bowman capsule. Okay? So in this situation, what are we going to see? Protein is 0. Why? Because proteins are removed in the process of ultrafiltration. Glucose will still be present. Urea is still going to be present and sodium will still be present. Now, let's take our third sample from this loop of Henle region. In the third sample, what are we going to do? Protein obviously is 0. But this time, glucose is also 0. Why? Because glucose has been absorbed back. The body has absorbed back the glucose in the first part of the tubule. Urea is now 3C. It's not C anymore. It's 3 times the concentration. Why? Because we have removed the glucose, we have removed the protein, we have removed a lot of water. So that, what does it do? It basically concentrates the urine. So the urea will increase in concentration. Sodium ions are going to be D. And last, let's take our sample from the collecting tubule. Protein is equals to 0. Glucose is equals to 0. Urea is so concentrated by the end of it, Viraj, that it's at 60 C. Sodium ions are, okay, so for sodium ions, there are 3 possibilities. Number 1 possibility is that that you have, if there's too much sodium inside the body will get rid of it. So you might see something like this. Or if there's not enough sodium in the body might absorb it back. So this is how the process of selective resorption actually works. And if we talk about the loop of Henle, you will notice that the loop of Henle is very important for resorbing water and the sodium and chloride ions. So loop of Henle really helps in concentrating the urine, right? Because this is where the water is resorbed back. This is where our ions are resorbed back. So this is where basically the urine concentrates. So in mammals that have very long loops of Henle, okay, they form really concentrated urine. So for example, desert animals like camels and all, they have really long loops of Henle so that they can absorb as much water as possible back into the body, and the urine is highly concentrated. In those mammals that maybe live in cold environment, they can have shorter loops of Henle. So loops of Henle also help in conservation of water, and they also help in concentrating your urine and, controlling the amount of body that is going to be excreted by the body. All right, Viraj, making sense? Any confusion regarding anything?
No.
Okay. So, all right, Viraj, I will go ahead and stop it over here for today. And in our next class, we'll go ahead and we are left with, we just have to go over our hormone, anti-diuretic hormone, which explains exactly how the body controls how much water to resorb back. And then we just have to go over how the body controls body temperature. So in our next class, we will go ahead and finish off with this topic. Okay.
Okay.
Okay. All right. Thank you so much, Viraj. I'll see you in the next class. Have a good day.
Bye.
Bye, Raj.